Genetics. All living things have different sizes, shapes, colors, behaviors, and so forth. You can see from the pictures in the background here. These are called traits. Human beings have many different traits. Some examples are hair, hair color, eye color, height, weight, blood type, face shape, personality, etc. Traits are determined by the genes that were passed down from your parents. The father of modern genetics is known as a guy named Gregor Mendel. And he was a monk that lived in Austria around the 1840s. He was inspired by fellow monks to study the variation in plants. Over a number of years, he conducted 29,000 experiments on pea plants. His discoveries went mostly unnoticed until the 1900s, many years after he died. So traits often come in two forms. As you can see from this picture, okay, we've got earlobes that are attached and earlobes that are detached. Okay, and that's basically what I say here. Okay, you have a gene that controls what kind of earlobe you have. Let's remember how this all works. The genes are in your cells. The genes are long strands called chromosomes. Okay, remember that they are within the nucleus of the cell. Remember back to mitosis and what that taught us about it. So what do you notice about these genes right here? We have 46 of them. Okay, you notice we have one from mom and one from dad, one from mom and one from dad. Okay, and if we have 2, 4, 6, 8, 10, okay, all the way through 22, that's 44. And then over here, these guys right here are 45 and 46. So our cells, our chromosomes have 46. Sorry, our cells have 46 chromosomes. All of our chromosomes are paired. Okay. Where did we get each one from? I already mentioned this. Okay, half came from mom and half came from dad. This means that genes are in pairs. Okay, so genes are chromosomes or on chromosomes. So remember those earlobes? You might have two attached earlobes, earlobe alleles, two dangly earlobe alleles, or one of each allele. But you'll definitely have two. Okay, that doesn't mean that you'll have one dangly earlobe and one attached earlobe. But what that does mean is that you will have two of what are called alleles, and those are the genes. Okay, so alleles. There are two versions of each gene. Okay, each version is called an allele. So depending on which allele you got from your parents, this affects the traits that you have. Dominant alleles, now this is really important. Dominant alleles are the ones that dominate or overpower the other alleles. Recessive alleles are the ones that are overpowered by the dominant ones. So what that means, and I'll explain it in more detail coming up here, but I like to think of it as this. We have the dominant one. Okay, you think of sports. You've heard of people say they dominated the other team, right? That means that they beat the other team, and they didn't just beat them, but they overpowered them. Okay, and so the dominant gene will overpower the other gene. It won't show the other gene. Okay, it won't show the recessive gene. And so therefore, the recessive gene is the one that's overpowered by the dominant gene. So now to define phenotypes and genotypes. Phenotype is the actual appearance of the organism, what it looks like. Okay, so we're talking about how it looks, how it acts, how it feels. Okay, these are things like our hair color, eye color. Okay, it might be a genetic disorder specific to a gene. Um, Genotypes, or a genotype, is the actual gene that an organism has as a part of the genetic code. So it's important to understand the difference between phenotype and genotype as we go through this part of the unit. Because some genes are dominant over others, the genes that you have are not necessarily expressed in your phenotype. What that means is that sometimes, let's say you have a dominant gene, say brown eye color. Let's say you have brown eyes, but your mom has blue eyes. Okay. And let's say that both of her parents had blue eyes, which means she could only give you blue eyes. So she gives you blue eyes, but because your dad has brown eyes and both of his parents had brown eyes, he gives you brown eyes, and brown eyes is a dominant trait. Okay. And so you get brown eyes. You don't get blue eyes because blue eyes is a recessive trait. So you won't express that phenotype of blue eyes. You will express the phenotype of brown eyes based on your genotypes. We always use capital letters to to show them. Okay, homozygous. If both, so these are we need to understand these terms as well. This is if both alleles of a gene are the same. So remember that the word in science, we look at words and we break them up. Okay, so the prefix homo means same, and so if you just remember that homo means same, okay, if both alleles are of the gene are the same, okay, that's homozygous. Okay, and the example that I give you here um, for okay, both are dominant for the gene, then we use uh, capital B, capital B. Okay, so that's for a dominant gene that's homozygous. We might also put a lowercase b. Okay, but that is if it's recessive right here. Okay, you can see if it's recessive, it's lowercase b, lowercase b. That's the only time that you'll see the phenotype. So if this was a person whose both parents had blue eyes, this is what their genotype would look like, and their phenotype would be blue eyes. Heterozygous is if the alleles of a gene are different. Okay, one dominant and one recessive. So you can see here, big B, little b is heterozygous. And again, hetero means different, the prefix. Okay, so hetero means different, and so that can help you to remember that we're talking about, if we're talking about heterozygous, we're talking about alleles that are different from each other. Okay, but sex cells are special when it comes to chromosomes and genes. We've learned this already. They only have one of each and not the usual two that each one of your cells has, those pairs. Okay, so why is this? Just a bit of a reminder. Okay, sex cells are designed to join together, and so, okay, 1 plus 1 equals 2. Okay, we can use these facts to make predictions about the features that children will inherit from their parents. So, and you'll see here, it's called a Punnett square. But let's pretend that a man with attached recessive earlobes and a woman with dangly homorecessive dominant earlobes have children. What kind of earlobes will the children have? Okay, well, this is how you do it. So you start. I want you to take out a piece of paper, okay, and go through this with me, so you understand how to do Punnett squares. It's crucial that you do this. So start by drawing, and pause the video as you go through, please. So start by drawing the Punnett square. So you draw this box, okay? This box with 4 boxes inside of it. Identify and add the parents with the genotypes. Okay, so the dad was homozygous recessive. So you'll put up here the dad on the top and the mom on the side. Now that is not necessarily the way it has to be, but let's, for our class and for our purposes, let's put the male on top and the female on the side, please. Okay, it makes it easier for my marking and also for continuity. Okay, so again, a homozygous meaning same. Okay, recessive dad, lowercase lowercase. Mom is homozygous dominant. Okay, so that's capital A, capital A. So now we need to start to cross the alleles with each other. So what we do is we take this A, okay, and this mom here would pass on an A here. Remember, she has 2 alleles, and this is why we have 2 for each. Okay, so she'd pass on one here, and she'd pass on one there. Okay, she'd also pass one on here, and pass one on here. There's a possibility of 4 offspring. Okay, then we cross with dads. Okay, so dad gives one, dad gives another. Okay, dad also gives them to these other children. Okay, this is now a complete Punnett square. These are the possibilities for these kids. Okay, from these particular parents. So remember, this is where the parents are. This is the dad, this is the mom, and these are the kids. Now, as far as percentage of a chance for offspring, if we have the whole box is divisible by 4, that means that this is 1/4 or 25%, okay, of a chance. Here's another 25%, 25, 25 for a total of 100. So depending on what you have in each one of these, you could have different chances of getting different alleles. Okay, there they just add up to 100%. Each square is worth 1/4 or 25%. So what is the possible genotypes? The possible genotypes here are big A, little a. That's the only one. Because 4 out of 4 times, these people will have a child with big A, little a. There's no way that they can have a little a, or a big A, because they're each homozygous dominant and homozygous recessive. Okay. Now, what does this mean? What are the possible phenotypes? Because those were the genotypes. So dangly earlobes, they'll have 100% of the time. Okay, why is that? Okay, well, it's because a dominant gene is present. Okay, this big A is present. Remember that the big A dominates the little a. Okay, and so the only way to show um, detached earlobe or attached earlobes is if you have a little a, little a. And by the way, the the letters are arbitrary. It doesn't matter what letters you use. Okay, I would discourage you from using an X, and you'll see that later when we get into sex-linked Punnett squares, or a Y for that matter. Okay, but anything else is fine. Attached earlobes, they will have 0 out of 4 times, or 0% of the time. Okay, so that is how you do a Punnett square.